Hello everyone. In this PowerPoint presentation, we will be discussing a skin disease in sheep called dermatophilosis as Group 80. Dermatophilosis is a dermatitis or an inflammation of the skin that is caused by a spore-forming bacterium called Dermatophilus congolensis. It is exudative, which means that a fluid emits from the scab formed due to the inflammation and it damages the hide of the sheep. It also sometimes results in death. The bacterium is filamentous and it's also a facultative anaerobe, so this means that it can survive with oxygen or without it. The bacterium mostly occurs in areas of high rainfall and humid conditions.
These are gram-positive bacteria that produce motile zoospores that invade the skin. They are pleomorphic facultative anaerobes that grow best at an atmosphere containing 10% carbon dioxide. They can also grow at a temperature of between 22 to 27 degrees Celsius. Optimum growth of the bacteria occurs on blood agar, brain heart infusion agar, and in tryptophan broth. In liquid media, its growth appears as a thick sediment. Transmission is by direct or indirect contact. Close contact is by rubbing each other when both animals are wet. Wet skin and wet crassulations are attractive to flies, which promote the transmission indirectly.
Weather plays a huge role in dermatophilosis as it is the most common in areas with high rainfall and tropical areas. Prolonged wetting by rain breaks down the natural protective barriers of the skin and allows the bacteria to spread. Rain and snow that wet hair coats cause matting and present the greatest opportunity for infection. The organism that causes dermatophilosis is found on the skin of the infected animal, but organisms can also be carriers of the disease but not show signs. The organism is able to survive outside the host in soil, but only in specific types of soil and water content. The soil pH does not influence its survival. It can survive for several months in soils, especially within cast-off crusts.
Animals naturally have barriers that protect them against penetration of microorganisms to their skin. When the barriers are compromised, it leads to the activation of dormant zoospores. When these are activated, they produce germ chips that develop into filaments, and the Dermatophilosis congolensis is able to penetrate the epidermis and establish infection. Where specific mechanisms facilitate the exit route. Regular dipping of sheep can initiate an outbreak of dermatophilosis or cause the spread of lesions on affected animals. Infected skin crust of carrier animals will become loose in the dipping tank fluid, and susceptible animals with existing skin lesions due to other causes or having macerated skin may on dipping become infected as well.
A swab of the affected skin, some fleece or a scab are suitable samples for diagnosis. Fresh scabs can be stored in distilled water and then analyzed by their concave base. Samples from the sheep's discolored fleece are helpful as they can be used for a differential diagnosis. This is because lesions can be a result of other bacteria found in the fleece that are not dermatophilosis. The correct transport method for these samples is essential to prevent contamination and to receive the most accurate results possible. This can be done by keeping bacterial culture from biopsy material or scabs in a clean envelope or a sterile specimen container. A special transport medium is not required for these specific samples. Smears are then prepared from the affected skin samples for analysis.
In terms of classification, D. congolensis is a gram-positive facultatively anaerobic bacteria compared to viruses, which are small infectious agents that are all obligate intracellular parasites. Fungi are eukaryotic multicellular organisms. In terms of morphological characteristics, D. congolensis has two major morphological forms, these being branching filamentous hypha and motile zoospores. Whereas viruses have four basic forms, and these are icosahedral, or helical, or complex, or binary, and all of them can either be enveloped or naked. Fungi have two major forms, and these are branching hypha or unicellular yeasts. When it comes to reproduction, D. congolensis releases sporangia in the presence of liquid, which then become flagellated and motile zoospores, and these are the infective form of the organism. Under favorable conditions, the zoospores form germ tubes that develop into branching filaments, and this is the invasive form of the organism. Viruses use the energy and ribosomes of their host cell to replicate as they have no organelles of their own. Fungi reproduce both sexually and asexually via spore production. In terms of genetic exchange, D. congolensis does this via conjugation, while viruses undergo subtle genetic changes through mutation, or they can undergo major genetic changes through recombination. Fungi undergo genetic exchange via meiosis during sexual reproduction. For D. congolensis, one can use stain smears with gram stain or aniline dyes and examine for the presence of branched filaments that are transversely and longitudinally septate. Viruses can be directly visualized via an electron microscope or indirectly via looking at the effect on embryonated chicken eggs and cell cultures. Fungi can be directly visualized through examination of wet preparations under a microscope. And when using staining, you can use lactophenol cotton blue for wet preparations and methylene blue or gram stain for yeast.